All right, hey everyone. If you've got a biology exam coming up, you are in the right place. Today we are tackling one of the most essential processes in all of life, bacterial DNA replication. Trust me, this is a core concept that is almost guaranteed to show up, and we're going to make sure you absolutely nail it. So let's just start with the big question, the absolute puzzle of it all. How does a tiny cell, like an E. coli bacterium, copy its entire genetic library, we're talking millions of base pairs, in less than an hour and make almost no mistakes? I mean, how is that even possible? That's the incredible molecular machine we're going to unpack today. I want you to think of this as your personal exam review session. We're going to hit the main concepts, meet all the key players, and, most importantly, I'm going to point out the common mistakes and the tricky questions that always seem to trip students up on exams. So, let's dive right in. Okay, so every story has a beginning. And for DNA replication, that critical first step is called initiation. This is all about getting the process started at the exact right spot on the chromosome and at the perfect time. Now that specific starting place has a name. It's called the origin of replication, or just ORIC. And here is a super important point for your exam. Bacteria like to keep things simple. They have just one single origin on their entire circular chromosome. You can think of it as the one and only starting line for this massive replication race.
So how does it actually kick off? Well, it all starts when these initiator proteins, called DNA, grab onto the ORIC. This binding actually bends the DNA, putting a strain on it and popping open a nearby region that's super rich in adenine and thymine bases. Now, why there? Here's the key. AT pairs are only held together by 2 hydrogen bonds, whereas GC pairs have 3. They're the weak link in the chain, which makes that spot much easier to pull apart. Once that little bubble is open, DNA helicase gets loaded on, and boom, we have 2 replication forks ready to zoom off in opposite directions.
All right, let's zoom in on one of those replication forks. See, a fork isn't just a place. It's a dynamic, incredibly complex piece of machinery. Honestly, the best way to picture it is as this bustling molecular factory with a whole crew of specialized proteins all working together in perfect sync.
And here it is. This diagram shows the factory in action. You can see all the key players right there, assembled where the DNA is being unwound. Each one of these proteins has a very specific job to do, and they all work together seamlessly. I know it looks complicated, but it's a beautifully organized system. Let's go ahead and meet the crew.
So let's break down who's on this factory floor. First up, you've got DNA helicase. Think of it as the demolition crew, just plowing forward, unzipping that double helix. Now, as it unwinds, it creates a ton of tension up ahead. That's where topoisomerase, also known as DNA gyrase, comes in. It's the tension relief specialist. It literally cuts and reseals the DNA to prevent knots. Meanwhile, the newly exposed single strands are held apart by single-strand binding proteins. They're the safety crew, making sure the strands don't just snap back together. And finally, you have primase. This is the foreman who lays down a little RNA primer, which is the essential starting block for the real building process.
Okay, listen up, because this next section is absolutely critical. The difference between the leading and the lagging strands is the heart and soul of DNA replication. And I can tell you, it is easily the concept that trips up students the most on exams. So let's break this down very carefully. The star of our show, the master builder of the factory, is an enzyme called DNA polymerase III. This is the workhorse. It's the one that actually synthesizes the new DNA. But it has 2 very strict, non-negotiable rules that it has to follow. And these 2 rules create a fascinating problem that the cell has to solve.
And here they are, the 2 big rules. Rule number 1, DNA polymerase can't just start from scratch. It needs that short RNA primer from primase to get going. And rule number 2, and this is the most important one to remember, it can only add new nucleotides to the 3' end of a growing DNA strand. This means it can only build in one direction, 5' to 3'. This one single rule is the entire reason we even have a leading and a lagging strand.
So with that 5' to 3' rule in mind, how does the cell copy both strands at once? Well, it uses 2 different strategies. For the leading strand, life is easy. Synthesis is continuous. It just follows the replication fork as it opens up, moving in the same direction. It needs one primer, and then DNA Pol III can just cruise. But the lagging strand, that's the tricky one. It has to be built moving away from the fork. So to solve this puzzle, it's synthesized in a series of short little chunks called Okazaki fragments. And each one of these fragments needs its own brand new RNA primer to get started.
This slide really gives us the big picture. And remember, we've got 2 of these replication forks moving out from the origin.
At each and every fork, you have one leading strand and one lagging strand being built at the exact same time. It's a really elegant, even if it seems complicated, solution to that fundamental 5' to 3' problem.
So at this point, the new strands have been made, but we are not done yet. Not by a long shot. That lagging strand is still this fragmented mess of DNA chunks and little RNA primers. So we need to call in a dedicated cleanup crew to come in and make it a perfect, seamless molecule.
The first member of our cleanup crew is DNA polymerase I. Now, it is crucial that you don't mix this up with polymerase III. Poly I has a special tool, a 5' to 3' exonuclease activity. You can pretty much think of it like a little molecular Pac-Man that moves along the strand and just chews up those leftover RNA primers.
But Poly is a serious multitasker. As it's chewing away the RNA primer in front of it, it's simultaneously using its other skill, its polymerase activity, to fill in that gap with the correct DNA nucleotides. It's the ultimate patch and repair specialist.
And here is a massive exam tip. You will almost certainly be asked to tell the difference between these two polymerases. So just remember it this way. Polymerase III is the main high-speed builder. Polymerase I is the specialized cleanup and repair enzyme for the lagging strand. Get those two roles straight in your head.
There's one last job for the cleanup crew. After Poly I has done its thing, there's still a tiny little nick in the DNA backbone between each of those fragments. So the final member of our crew, DNA ligase, comes in and acts like molecular glue. It creates that final bond, sealing the nick, and joining all the Okazaki fragments into one continuous, perfect strand of DNA.
Okay, so we've talked about the incredible speed of this whole process, but what about accuracy? I mean, a mistake in your genetic blueprint can have really serious consequences. To prevent that, the cell has built in multiple, redundant layers of quality control.
But before we get to proofreading, let's ask a simple question. Where does all the energy for this building come from? Incredibly, it comes from the building blocks themselves. Each new nucleotide that shows up for work arrives as a triphosphate. When DNA polymerase adds it to the chain, it snaps off two of those phosphates. The energy that's released from breaking that high-energy bond is what powers the whole reaction. It's an unbelievably efficient system. So this incredible accuracy comes down to three main checkpoints. First, you just have simple base pairing rules. You know, A with T and G with C are just chemically more stable and fit better than the wrong pairs. Second, the enzyme itself helps out through something called induced fit. The part of the polymerase that does the work actually changes shape to perfectly grip a correctly paired nucleotide so it rejects the wrong ones. And finally, if a mistake still somehow slips through, there's an active proofreading mechanism.
And this proofreading function is just amazing. If DNA polymerase accidentally adds the wrong base, it can actually feel the mistake. It creates this little physical bump in the double helix. The enzyme then hits the brakes, shifts the end of the new strand to a different part of the enzyme, the 3' to 5' exonuclease site, and just snips out the wrong nucleotide. It is literally a delete key that allows the polymerase to fix its own typos as it goes along.
We've now gone through the entire process, from kicking things off all the way to quality control. So for a final review, let's just do a quick hitter summary of all the key players. You can think of this next slide as your ultimate cheat sheet for the entire replication factory.
And there it is, the complete cast of our replication factory. You've got helicase to unwind, topoisomerase to relieve tension, primase to get things started, polymerase III as the main builder, polymerase I to clean up those primers, and finally, DNA ligase to seal everything up. Go ahead and pause the explainer here, and just make sure you know the specific job of every single one of these enzymes. This table right here is pure exam gold.
So let's just end on this thought. We've been calling this a coordinated factory, but there's no boss, there's no one shouting orders, no central brain controlling it all. It's a self-assembling, self-correcting machine that runs purely on the laws of chemistry and physics, all shaped over billions of years of evolution. It is just an absolutely stunning piece of molecular engineering. Thanks so much for watching, and seriously, good luck on your exam.